So I'm going to talk about neurons that make you move. And when I put this title, and you have just witnessed this incredible music, people often think that I'm going to talk about these really amazing movements that you just saw or hear, or Simone Biles doing her flips. But I want to break your bubble. The hard truth is that most of neuroscience right now struggles to understand how the clumsiest specimens of our species can just walk around and not fall over. And hundreds of you just walked inside this room. You stopped in order to avoid bumping into someone in front of you. You twisted and turned, and you took your seats. And there is no robot that can do that right now. And the reason for that is we just don't understand how our brains control movement that gracefully. So when you walk, the muscles in your hands and legs are contracting and relaxing, and that's actually done by your nervous system, which is your brain and your spinal cord, that consists of neurons that are continuously talking to each other with these little flashes of light that you see there. And these neurons talk to each other at these nanoscale units called synapses by secreting chemicals. And all of this is ultimately encoded in your DNA. So if you really want to understand how we behave in general, or how we walk, you have to take this multi-scale view of things. And that's important because defects in your DNA, which are genetic disorders, lead to impaired neural circuit function, which ultimately impairs your movement. We have billions of neurons and trillions of synapses. How do you go about figuring out how this circuit is put together for this graceful movement? And fortunately, all animals walk, and they walk in a very similar way. And the reason for that is all of them have evolved to walk on this same planet, right, with its special terrain and its gravitational field. And even their legs look very similar. So this is like a leg of a fly and a leg of a cat. And they both have a knee joint, which does the same thing, which is controlled by similar muscles. A fly can do this with 140,000 neurons. A mouse can do this with 70 million neurons, which is a fraction of neurons compared to humans. Which is why most of science is focused on using these as model systems to understand how brains and spinal cords control movement. And that is a much more approachable problem at this moment. And as David mentioned, in my lab, we mainly focus on using this fruit fly as a model system, although we are about to start some work in the other model system, mouse also. But the reason why we use this particular fly and not just any insect, which fits the same criteria, is because of this guy. So Thomas Morgan was awarded the Nobel Prize in 1933 for his work on uncovering the basis of genetics and hereditary. And he decided to use a fruit fly back in the day, in the early 1900s. And the reason for that is these flies are really easy to grow. They're just in our backyards. And they grow really fast. So from an egg to an adult fly, it just takes 10 days. And so it's great for studying genetics. And Morgan actually went on to map the first mutation ever that was done by humans to the genome of the fly without even knowing that the genome is made of DNA. So that shows the power of the model system. And of course, a lot of people followed in his footsteps, and there have been fundamental discoveries in biology, all the way from genetics to immunology to neuroscience, that were actually possible because all of these people were working in fruit flies. And this is just the list that got the Nobel Prize, but there's a lot more, of course. And the reason for that is because flies share a lot with us. They have the same systems, the same organs. almost 75% of disease-causing genes identified in humans have homologs in flies. They have the same neurotransmitters. So if you give a sleeping pill to a fly, the fly sleeps. If you feed her coffee, she wakes up. And the reason for that is because all these substances interact with the fly's brain in the same way they interact with your brain. So if you understand something about how a fly brain works, you can hope to understand something about our brains. So if this is a mouse brain and a fly brain and their spinal cords next to each other. And I'm just outlining sort of the broad circuit scheme for walking control. So this is the brain stem that sends commands down into the spinal cord, which controls your walking movements, which is sort of important for our work. And it sits at the base of the brain, both in the fly and the mouse. And so that's sort of the area that we are interested in. But there's in 2025, there's another reason why this is a great model system, and that is this. So a few months ago, a large consortium of neuroscientists spanning continents and universities, including my own lab, published this wiring diagram of the entire fly brain at nanometer resolution. So this, in fact, is the biggest wiring diagram of any brain known to humans right now. And this is mapped to synaptic resolution. So here you're seeing some fragments of this connectome, as we call it. So these are sort of all the neurons that sit in the optic lobe of the fly. So these are visual projection neurons that take information from the eyes to the brain of the fly. These are neurons that take information from the central brain back to the eyes, all of them. These are all the central neurons in the brain that the fly uses to think and decide. And these are all the ascending neurons that take information from the spinal cord up to the brain. And then these are descending neurons that take information from the brain down to the spinal cord. And sort of a similar map has been done for the sort of spinal cord, which is called as the nerve cord of the fly. So now we have all these tools. So we're going to answer how George walks. But seriously, these are the three sort of fundamental questions that we are after, which unfortunately we still don't really understand. So how is a basic walking pattern generated? And how is a pattern modified in order to turn and stop and change speed? And how to do all of this without falling? So there are like 140,000 neurons in the brain of the fly, another 30,000 in the nerve cord. How do you go about finding which ones are important for walking? So this is where genetics comes in. So because of this amazing guy, there is like more than 100 years of tool development in fly neuroscience or fly biology in general. And because of that, we have really powerful genetic tools to manipulate individual cells in the body of the fly. So here I'm saying a genetic tool that allows us to play with a single neuronal type out of all these 140,000 neurons in the brain. I could use this tool to turn this neuron green by expressing a green fluorescent protein. And then when I look at the brain of this fly under the microscope, everything else is colorless, which is, I've colored it pink because we have stained for it. But everything else is colorless except this one particular cell that's been turned green. And instead of turning a neuron green, you can do a lot of other things with it. You can express this light-sensitive protein in this neuron that's normally present in an algae and uses it to move towards light. We have a version of this protein that responds to a particular wavelength of red light. We express this in our favorite neurons called P9, and then these neurons become sensitive to light. So now if you shine light on these neurons, they start firing these action potentials, which are represented as these dots over here. So now this is actually a real recording of a P9 neuron as we shine light on it. So if you stick an electrode into this neuron, you can actually see the potential changing. And this is what we have plotted over here. So when the light comes on, the cell becomes like more positive, and it starts firing action potentials, which are represented by these spikes over here. And we can actually pulse this light at different frequencies to make the neuron fire at different frequencies. So what we have actually is a very sophisticated remote control for the brain of the fly. So we can literally do this for any neuron. And the beauty of this is that this red light that we use can actually pass through the fly. The fly doesn't even see it. It's beyond its visual reception. And you can just have a red flashlight, shine it on the fly, and every cell in the fly's body will be exposed to this red light, but all those cells will be insensitive to it, except your favorite neuron, which you have genetically engineered to become sensitive to light. So this is what happens if we do this in a real fly. So this is a fly just chilling away, and then a red light comes, and the fly starts turning. And you don't see the red light here because we put filters on the camera that block it. But here I'm activating this right P9 neuron or the left P9 neuron, and the fly starts turning right or left. So you should appreciate that the fly is sort of minding its own business before the light comes on. And when the light comes on, it just stops doing whatever it was doing and now decides to turn because that's what its brain is commanding it to do. So now if you zoom into this fly and you see really what's happening at the level of legs, you can appreciate that every leg sort of starts stepping in a slightly different way. So these are like the footprints of the fly, the front legs, middle legs, and hind legs, and all of them are doing slightly different things. So the question is, how does a single neuron control all these 6 legs? And in fact, even the neck of the fly moves. And so you have to control all these muscles in all these joints, and how is this one cell doing it? So the wiring diagram actually tells us that before this cell actually exits the brain, it recruits this whole population of neurons that project to different parts of the spinal cord of the fly and is sort of poised to control different aspects of leg movements. And we have actually now found that you can represent that wiring diagram in this sort of a plot where the P9 neuron sits on the top, and all these other neurons that it controls in the brainstem sit in this layer. And then each of these neurons controls slightly different aspects of leg movements. And together, they're sort of guiding the fly. And this sort of loosely resembles what a puppet master does when he controls a string puppet or marionette. But so in this case, P9 is basically the puppet master, and I'm playing God because I'm controlling P9. But if you look at, I was reading up on this, and it takes a while to become a professional puppet master. You have to train for like 5 to 10 years. And if you look at this professional puppet master, he has these white rods over here that actually couple movement of some joints together, because the puppet master knows that when one joint goes up, another has to come down, or some joints have to go up and down together, otherwise it would just look unnatural. And that's what we think all these connections are doing. So this layer is sort of coupled to each other through all these connections that are represented by these arrows over here. And so our hypothesis is that the nature of this wiring diagram is actually the reason why the fly performs this graceful turn and doesn't fall over. And we are investigating that in more detail now. So this tells us a bit about how a walking pattern can be changed. But how does a walking pattern start, and how is it generated? And for that, we are using another neuron that I discovered back in my postdoc days. So this is a fly that just starts sprinting straight forward when the light comes on. We call these the Bolt neurons. I named them the Bolt neurons after Usain Bolt, since the fly sprints. the cool thing about these guys is that, whether you activate the left neuron or the right neuron, the fly just goes straight. And if you kind of look at the high resolution leg movements, you see this beautiful coordinated forward walk. So this is the perfect starting point for understanding pattern generation. And in a similar way to that previous work, we have figured out all the strings that are pulled by the bolt neuron. And I'm just showing a few of these downstream connections over here. And in fact, this yellow neuron over here can also make the fly walk forward when you activate these neurons. But if you look closely, you'll notice that this fly doesn't have a head. And that's insane, right? Because what we have found is that even if you just stimulate the output of this neuron in the spinal cord, it's enough to generate walking. And that tells us that all the pattern generation machinery is localized to the spinal cord of the fly, just like it is localized to the spinal cord of a human. And now we, in fact, have a circuit that we can use to figure out how patterns are actually generated. So I told you a bit about different types of neurons that are active when you walk, and there are a lot more. So the next question we asked is, what happens when you stop walking? Do all of these neurons suddenly become silent, or are there specific neurons that need to be inhibited? And we had, again, similar tricks to identify a bunch of neurons that cause the fly to stop walking when we stimulate them. And then we actually, in sort of similar analysis, figured out that the fly uses two main strategies to stop walking. And the first one is actually sort of like pulling your foot from a gas pedal. So this is the P9 neuron and all its downstream neurons, and this is the bolt neuron and all the brainstem neurons that it recruits. And we found that this stop neuron, it inhibits only specific nodes in this layer. So not all neurons that are downstream of these sort of puppet masters are equally important. So there are specific ones like this BDN2, which causes walking in the headless fly, that are more important than others, and we think they are the gas pedals of the fly brain that need to be inhibited in order for the fly to stop. But then there's another mechanism which we identified serendipitously. We were not even thinking about it, but we found this another class of neuron that actually causes stopping by just increasing resistance in all the joints of the fly. So it actually directly controls the activity of muscles. So this is the knee joint of the fly, just like your knee joint. And there is specific activity in the muscles that happen that resist the movement, just like a brake resists the movement of a wheel. And that's why we named it the brake neurons. So towards the end, I want to sort of reiterate our fundamental sort of goals that we are after. So of course, the main goal of my lab is to uncover the basic principles of walking. And we are using all these amazing tools for that. We are also exploring if these strategies are conserved across animal species. So we are collaborating with a mouse lab for that. And some serious folks in the audience might look at this and think, you guys are just having too much fun. You are just playing in your lab every day. And to them, I want to answer yes, we are. We are having ridiculous amount of fun, and that's essentially the reason we wake up every day and go to the lab. And most of the breakthroughs that happened in science, like everything from invention of calculus to discovery of antibiotics, was because those people were having fun and playing around. And that's why it's curiosity-driven research that's the motto of Max Planck is so important. But of course, we are interested to know if we can apply what we learn to help others. And like I told you, impact in your genome can actually lead to defects in your behavior. And we recently published this paper with our collaborator Seth Tomczyk, where we took a fly model of a human disease, and we mapped out the motor control aspect of it. And now you can realize that once you know a lot about how a system functions normally, you have this special power to spot what goes wrong. And so we are using that now. In future, we are hoping to use it to even screen for drugs. And of course, the last bit is what David was alluding to. It's about inspiring the future of neuroscience. And at Max Planck, we have this Brain Exploration Day where people can come and look at all these amazing light-activated flies. We also have the Ask Max van, as David mentioned, and our education outreach people actually visit different schools in the county. And this is actually from last week. So Carla visited Eagles Middle School, and we got like this deck of thank you cards from them, from the students. And one student actually wrote this, which I found really interesting. So looking at fruit flies and how they reacted to light was really interesting. And he goes on, but he ends his thank you card with this. I wish you were here for a little longer to share everything you knew. And that tells us that we have sparked the curiosity in this kid. And if even a few of you feel like this after my talk, then I've done my part. And you're always welcome to come to Max Planck and learn more. And with that, I want to acknowledge all the resources that we have and this amazing team of scientists that work in my lab and actually feel the same way about doing science as I do, and also funding. And thank you for coming here.